Welcome back. So now that we understand how proteins are made by using the information encoded in mRNA, the next question becomes, where do the mRNAs come from? Please take a moment to review the learning objectives for this lesson. While translation is the biological process of making proteins from mRNA, transcription is the process of making RNA from DNA. In our cells, the permanent repository of genetic information is our genomic DNA. Like mRNA, DNA is made up of long chains of nucleotides. In the case of genomic DNA, these chains can be hundreds of millions of nucleotides long. Each chain is tightly packed into a structure we call a chromosome. Chromosomes reside in the cell nucleus and contain all the information needed to create and maintain a functioning human. But each cell type only needs to access just a tiny fraction of that vast information library to make its specific set of proteins. So when a cell needs to make a particular protein, it copies that bit of genomic DNA into RNA via a process called transcription. The protein that makes these RNA copies or transcripts is called RNA polymerase. The initial RNA transcript goes through a series of maturation steps, which we won't discuss here, to generate an mRNA. This mature mRNA is then transported out of the nucleus and into the cytoplasm, where the process of protein translation occurs. This is a one-way trip. Once the mRNA is in the cytoplasm, it cannot go back into the nucleus. This flow of information from DNA to mRNA to protein is known as the central dogma of biology. DNA, the permanent repository of genetic information, is transcribed into mRNA, a transient copy of just the information needed to make a single specific protein, and the information in the mRNA is translated by ribosomes into proteins.